Myself, Dr.S.Sowmya, Assistant Professor, Clinical Laboratory Technology, Arignar College, Krishnagiri. My topic is about chemistry of carbohydrates. Let's see what is carbohydrate. Carbohydrates are polyhydroxy, aldehyde or ketone derivatives. It is widely distributed in plants and animals. Usually plants are rich in carbohydrates than animals. It is very essential to maintain life in plants and animals to provide energy. And carbohydrates, it classified into three groups. Monosaccharides, it is a simple sugar, contain only one sugar molecule, can be broken into simpler units on hydrolysis. And oligosaccharides, it means oligo means few. This compounds sugar yield 2 to 10 monosaccharide units on hydrolysis. And then polysaccharides, poly means many, also called glycans. This compound sugar yield more than 10 monosaccharide units on hydrolysis. Disaccharides. Disaccharides formed by the union of two monosaccharides held together by glycosidic bonds. And properties are sweet in taste and water soluble. Some sugar molecules are classified into two types, that is reducing sugar and non-reducing sugar. Reducing sugar donate electrons to other molecules and reducing them, for example glucose, fructose, lactose and maltose. And non-reducing sugars can't donate the electrons to other molecules, no any free aldehyde or ketone groups, for example sucrose and terrahalose. And the disaccharides of maltose having two glucose molecules, lactose that contain glucose and galactose. In non-reducing sugar, sucrose contains glucose and fructose. Maltose, it is a two units of glucose joined together with alpha 1,4 linkages and broken down into two glucose molecules on hydrolysis. In biological system, the action done by the enzyme of maltase and its properties are soluble and sweet in taste, and it mainly present in germinating seeds. Lactose. Lactose consists of galactose and glucose bonded with beta 1,4 linkages. It broken down by the enzyme of lactase. It is present in milk. And then sucrose, it consists of alpha D glucose and beta D fructose joined by alpha 1,2 linkages. It broken down in the intestinal enzyme of sucrose. It is present in sugarcane, beetroot and pineapple and carrots. Then cellobiose, polymer of two glucose units linked by beta 1,4 bonds. It is hydrolyzed by the bacteria and it is indigestible by the humans. And polysaccharides. Polysaccharide is a repeat units of monosaccharides. It classified into two types, that is homopolysaccharide and heteropolysaccharide. Homopolysaccharide, it means same. It divided into two types, that is storage polysaccharide and structural polysaccharide. Storage polysaccharides are starch, glycogen, dextrose, inulin, and structural polysaccharides are chitin and cellulose. Starch, it exists in two forms. One is amylose and another one is amylopectin. That starch is synthesized by a process called photosynthesis in plants. It is present in wheat, rice and potatoes. And amylose, it is unbranched having 250 to 300 glucose units joined by the alpha 1,4 linkages. It twists into helix with 6 glucose units per turn. And amylopectin, it is highly branched structure having approximately 80 branches, where mainstream has alpha 1,4 glycosidic bonds at the branch point at alpha 1,6 linkages. Then glycogen, it present in animals, hence it is called as animal starch. It is a branched in structure joined by alpha 1,4 glycosidic linkage at branch point by alpha 1,6 linkages, glycosidic linkages. And then chitin. Chitin is closely related to cellulose, having beta 1,4 linkages between beta D glucose units. It is found in fungi, carbs, and insects. Then cellulose. Cellulose is found in cell wall in plants and provide a physical strength. It is structural it is a structural polysaccharide. So it has a glucose units linked by beta 1,4 glycosidic linkages. In humans, cellulose is not digested since man doesn't have the cellulase enzyme. Thank you.